Hello, folks. Welcome to AP Biology and our notes on the cell structures and their functions, essentially. We're going to cover half of the unit deals with that, and the other half of Unit 2 deals with cell membrane and cell transport, how the cell moves things in and out. And I thought we'd give it a try here with me doing a video as opposed to watching the pre-produced ones or doing live notes. So let's go ahead and get started. I do recommend you take notes on this. I do have the notes posted on Classroom, so feel free to pull those up and follow along with those or take notes on those, or you can do it in your own notebook and just pause when you need to. So this first section, guys, deals with what I'm calling 2.0, which is cell theory, and 2.1, subcellular components, which is basically just what are the organelles that we're looking at in AP Bio, and section 2.2, those structures and what their functions are and how they interrelate. Cell theory in our book is covered on page 93, and our book kind of combines 2.1 and 2.2 and talks about the organelles that we're looking at from pages 102 to 112. So when you're doing your reading, those will be the sections to pay close attention to go along with this section of the notes. Let's take a look at cell theory, guys. Okay. We need to know about cells because cells are the basic structural and functional unit of life. In other words, all organisms are composed of cells. And if you look at our last bullet point here, if we want to understand how our different tissues and organs function, that depends intimately on how our cells function as well. Everything that happens at a tissue or organismal level has to happen, at least in part, within specialized cells within our body. So the structure of that cell determines how it functions, and the structures of all those cells and their functions determine how those tissues and organs function. Right? All cells come from other living cells. Okay? And that goes along with basically, if all cells come from other living cells, all cells present on planet Earth right now, as far as we know, come from a continuous line of descent from the very first cells that appeared on this planet way back when, which is kind of mind-boggling to think about, over what, 3 billion years ago? Pretty crazy. And cells are the smallest living things. Just a couple of reminders. What do cells need to do every day? Cells need to build proteins every day so they can carry out all their functions. They need to get energy and convert that energy so they can carry out those functions. And they need to make more cells. Otherwise, life would end. So those are three things they need to do. So these organelles that we're going to be looking at in these cell functions look at through the eyes of making proteins, getting energy, and making more cells. All right. So here we can see just a general picture of the organelles within a standard animal eukaryotic cell, which is kind of what we're going to focus on. There's some differences when you get to plant cells and some bigger differences when we get to prokaryotic, but we'll take a look at those later. Let's focus on this for now. All right. The cytoplasm. Now, in biology, you might have thought the cytoplasm was all of the liquidy stuff in the cell. We simplified it a little bit there, but the cytoplasm really is technically all the material between the plasma or cell membrane and the nucleus itself. So that includes all the stuff floating around in there, the organelles and everything. The cytosol is what is the watery component. So it's water with dissolved protein, salt, sugar, and other solutes in it. That's cytosol. Then we have the cytoplasmic organelles, the organelles floating in there, okay, the things that carry out the metabolism of the cell. And then there's other things which we call inclusions, different chemical substances like pigments and bits and pieces that are too big to be included as part of the cytosol because they're not dissolved in there. Okay. Let's start taking a look at these organelles. We have special cellular compartments. So the cell is compartmentalized into different pieces and things that are separated by membranes. And the fact that they are compartmentalized means they can specialize and they can be more efficient at getting things done. We'll talk about that more a little bit later. Some interesting membranous ones like mitochondria, lysosomes, the endoplasmic reticulum, and Golgi apparatus. Some non-membranous ones would be things like the cytoskeleton, centrioles, and ribosomes. And for AP biology, it doesn't seem like they talk too much about cytoskeleton and centrioles, so we won't. But we will focus mainly on ribosomes. All right, so let's take a look at ribosomes. Now, ribosomes are present in all cells. So whether you're prokaryote or eukaryote animal plant, fungi, protist, they have ribosomes. Okay? These ribosomes are little granules made up of rRNA, which is ribosomal RNA, and proteins. Okay? And if you remember from biology, really what they do, they make proteins. They take the information that was coded in the DNA that then gets carried out to the ribosomes on messenger or mRNA. The ribosomes read that information and use it to put the right amino acids together in the right order to make a polypeptide, a protein chain. Okay. The ribosomes can be found in two different places in a cell. They can be free floating. Okay. And the free floating ones are just kind of floating around in the cytosol, number 3 on this picture here. They synthesize soluble proteins that are used within the cell itself. So proteins that are just floating around within the cell, soluble in that cytoplasm or the cytosol, those are made in the free floating ones. Okay. The rest of them are membrane bound ribosomes. And those ones are stuck to membranes, most specifically onto endoplasmic reticulum or rough ER membranes, which we'll talk about a little bit later. And their job is to synthesize proteins. Synthesize means to make again. To synthesize proteins that get incorporated into membranes within the different organelles of the cell, and ones that are going to be exported out of the cell to get into interstitial fluids or the bloodstream or wherever, so they can go to other cells and do other jobs. Okay. And like I said, that leads us to the endoplasmic reticulum, which is one of those fun words to say if you want to say it with me. Endoplasmic reticulum. If you enunciate, it sounds pretty cool. Endoplasmic reticulum. And these are a bunch of interconnected tubes and membranes. They're attached to or contiguous with the nuclear membrane, the nucleus. And there's two varieties, rough ER and smooth ER. As you can see in this picture, we got the pinky purple is the nuclear envelope. Then attached to that, we have the ERs in blue. Rough ER is studded with the ribosomes. That's why we call it rough. And that affects what its function is because it's got ribosomes on it. And the smooth ER is called smooth because it doesn't have that on them. So it doesn't look rough under high magnification. And that means it does different functions because it doesn't have that same structural component. The rough ER helps to compartmentalize the cell. It's very involved in making these separate organelles, or at least the membranes for them, get made in the rough ER. Its external surface, like I said before, is studded with ribosomes, and it also manufactures all secreted proteins. All proteins that are going to be secreted out of the cell get made in the rough ER. It's also mainly responsible for the synthesis or production of integral membrane proteins and phospholipids for cell membranes. So it is a big part of making those membranes that form the cell membrane and the membranes around these various organelles we're going to talk about. And if those membranes have proteins that need to be embedded in them to do certain functions, like synthesizing ATP or allowing certain molecules to diffuse through the cell membrane, they will be made here on the rough ER. The smooth ER, these are tubules arranged in kind of a looping network, and they catalyze or speed up the following reactions in various organs of the body. Lots of different things. Now, for AP Bio, you don't need to know all the different specific things they do in different specialized cells. Because if we're looking at the smooth ER in bone cells, osteocytes, they do different things than the smooth ER in liver cells. Okay. So, just for example, in the liver, lipid and cholesterol metabolism, along with the breakdown of glycogen, kidneys, and detoxification of drugs. Detoxification, right there, that is something you should remember for smooth ER. It is involved in detoxification, okay, in the liver and within cells themselves. There's oftentimes materials get into the cell that you need to get rid of, and smooth ER helps to do that. I also highlighted lipids because if you're dealing with lipids and lipids only, you're probably dealing with smooth ER. If you're making or breaking down lipids, you're dealing with the smooth ER. In the testes, synthesis of steroid-based hormones, and steroids, if you remember, are a type of lipid. So you can see that there. In interstitial cell, we're dealing with the absorption, synthesis, and transport of fats. Once again, a lipid. Okay. In skeletal and cardiac muscle, it actually helps with the release of calcium. So it has lots of different things, but some commonalities that you'll see is it's dealing with lipid metabolism and detoxification. Those would be the two big takeaways here. Now, moving out from the ER, we get to the Golgi apparatus, kind of stacked, flattened, membranous sacs. Some people say it looks like a stack of pancakes from the side. If we want to generalize it, we say they function in the modification, concentration, and packaging of proteins. Okay? They're going to gather these proteins in. They're going to modify them. They can concentrate the proteins in other molecules and then package them up to ship them either elsewhere within the cell or out of the cell. So to get a little more specific, the modification part, they correct folding and modification of new proteins. So proteins, remember, they have their primary shape and then they have to fold up into their secondary and tertiary and quaternary shape. Some of that can happen in the cytoplasm, some of that happens in the ER, but there's enzymes within the Golgi which can help correct that, can unfold and refold things to make sure they're folded properly. Packaging and distribution of materials to different parts of the cell. So it'll take this stuff in, package up bundles of them, and then send them out elsewhere within the cell. Synthesis of cell wall components, if we're talking about plants or bacteria or fungi or protists that have cell walls. They produce lysosomes, which we'll talk about in a little bit, that break down materials. Now, the way Golgi apparatus works, transport vessels, which would be little sacs with phospholipids around the outside, double membrane phospholipid sacs, from the ER will fuse with the cis, or what we might know, the cis face of the Golgi apparatus. Cis face means it's the near face, the face near to the ER. Okay. Those proteins will then pass through the Golgi apparatus and do whatever modification and packaging need to happen to the trans face, the far face of the Golgi, the one closest to the cell membrane. Then it'll butt off little secretory vesicles, once again surrounded by a double phospholipid membrane, and move to designated parts of the cell or out of the cell. And the reason these vesicles can fuse with and then butt off of each other is because these cells have phospholipids. Now, the polar phosphorus heads of those phospholipids like to line up, and they will be able to bond with the polar heads of the organelle that they're getting to, or the cell membrane, so you can have them kind of fuse together and become one. We'll do a little video of that here in a little bit. And the nonpolar tails, the hydrophobic tails, will also be able to interact together. And basically, the two membranes will become one. So we'll show a little video of that in a minute.
Membranes will only fuse together when they are brought within a few nanometers of each other. This process is accomplished by proteins called snares. Different forms of these proteins are bound to particular membranes and provide additional target specificity. Once the vesicle is docked, a V snare anchored to the vesicular membrane and two T snares anchored to the target membrane coil together. In all cases of vesicle fusion, this coiling of snare proteins tightens the gap between vesicle and target membranes, squeezing out any water in between them. Snare coiling releases the energy needed to overcome the repulsion between the polar heads of the two membranes. When the membranes get close enough, hydrophilic heads from each membrane spontaneously flip directions, causing the closest layers of the two membranes to merge together. Spontaneous flipping is the rate-limiting step of vesicle fusion because it forms an unstable, hemifused membrane. Because hemifusion is highly unstable, it quickly rearranges to form a fully fused membrane. Once the vesicle has fused with its target membrane, the cargo is released into the luminal side, and membrane-bound proteins in the vesicle become part of the target membrane.
But here we can see kind of this whole system. So we got, coming into the cis face of the Golgi, we have a transport vesicle from the rough ER. Fuses, the membranes fuse here, so it can dump in its contents. They get modified and packaged. It can butt off, and the transport vesicle leaves, and it can go elsewhere in the cell or out of the cell. Say, make a lysosome. I don't know if any of these have lysosomes here. Not in that one. So we can kind of see this whole process here. So we got, if you want to think through the process of making something that's going to be excreted from the cell, you can have mRNA comes from the DNA, is made from the DNA, from a gene on the DNA, comes out through nuclear pore to one of these ribosomes on the rough ER, which will make that protein along with any attaching it to any lipids that needs to get made to, works its way through. That new protein can then come via transport vesicle to the cis face of the Golgi, can enter into the Golgi, be modified and packaged, and then secreted on out here. And we've now left it out here. And in addition, we've made the cell membrane a little bit bigger, and that promotes growth. And we can see the same thing here again. Oh, here's one where we've made actually a lysosome. And we can see how the cell membranes fuse with the secretory vesicle membrane, and all this material can come out via exocytosis, which we'll talk about in another section of this very unit. All right, let's take a look at the mighty mitochondria. I'm sure heard of this. It's the powerhouse of the cell, right? And what cellular process happens in the mitochondria that provides energy for our cells in the form of ATP? What is that process known as? Yeah, aerobic cellular respiration. In biology, we learned that we get things like glucose, those covalently bonded glucose molecules. There's a lot of energy stored in those bonds holding the glucose together. If we can break those bonds, we can convert it into a type of energy, chemical energy that the cell can use more easily, called ATP, adenosine triphosphate. The first part of that, breaking the glucose molecule in half, can take place in the cytosol of the cell. But then we got these two hunks, and we can produce a couple ATPs. Then we got these two leftover hunks of glucose with lots of bonds still holding them together. Those are what get into the mitochondria, and in the mitochondria, then we can further break down those bonds and release that energy and convert it into a whole bunch more ATP molecules. So in total, you can get maybe 2 ATP molecules from that first bit of cellular respiration, in the cellular respiration in the cytosol, called glycolysis or anaerobic respiration. And then in the second half, we can get 17 times more energy. So 34 more ATPs from breaking down the rest of that glucose molecule, for a total of about 36 if you go through the whole process of aerobic cellular respiration. And don't worry, we will get into cell energy. There's a whole unit on that later, but just a little reminder from what we've learned before. Now, the mitochondria itself has a double membrane structure, and inside it's got a big folded set of membranes called cristae, and those folds greatly increase the amount of surface area of those membranes. Okay, if you recall, if we just have a big old balloon, this is all the surface area that's in that balloon. If we can fold up a whole bunch of stuff inside that balloon, we can get a lot more surface area. Okay, and that is important. We'll talk about that in a minute. So here's the question. So here we have a mitochondria. We can see the outer mitochondrial membrane, and then the inner one with these folds called cristae. And inside there, we've got this fluidy stuff called the matrix. You can also see that on that cristae membrane, that inner membrane, we have a bunch of enzymes stuck to it. That structure is extremely important. That increase in surface area is extremely important for the inner folds. I want you to pause right now and think about, why is that so important for the function of the mitochondria? Yeah, increased surface area means we can pack a lot more of these enzymes that are going to promote cellular respiration into a very small area, so we can be very efficient at it. If we didn't have all those folds, it would take up too much space. It would be too inefficient. But by folding it all up in there, we can increase the surface area, but keep it in a combined smaller volume, and the cell can be much more efficient that way. Once again, compartmentalization within the cell, which is important, we talked about before, and increased surface area are two key factors you're going to see in cells and just in systems in general as you go through biology. Okay, so a little bit about where different parts of aerobic cellular respiration happen within the mitochondria. I'm just going to introduce it here. Like I said before, we are going to get more in depth into what these different things are in a different unit. But since we are talking about the mitochondria's structures and their functions now, we do need to introduce what those functions are. We'll get detailed into how those functions work in a different unit. So in the matrix, this liquidy stuff in between the cristae, we have the Krebs or the citric acid cycle. The electron transport chain and AT synthesis occur in the mitochondrial membrane itself, and those are catalyzed by these enzymes we see in the inner membranes. So that's where the extra surface area comes into play. Next, let's take a look at lysosomes. Okay. Lysosomes, the prefix lys means to break apart. Okay, we talked about that with hydrolysis or water cutting in the last unit, where we break down our organic molecules is broken down by adding in a water to break the covalent bond holding these various monomers together into the polymers. So lysosomes contain digestive or hydrolytic water cutting enzymes, so they help to speed up hydrolysis, and they contain them in there. They're little digestive sacs, and we use them when we need to break things down within the cell. So if there's bacteria or viruses in there or toxins we need to break down, the lysosomes will engulf them and break them down. Okay, we can see that down here in this picture with what we call phagocytosis, where we can take a food vacuole comes in, the lysosome comes over to that food vacuole, releases its enzymes in there, and we can digest that food into smaller pieces that we can then reuse for other things, or take to a mitochondria so we can get the energy out of it. We can break down non-useful tissue and non-functional organelles. We see that over here, where we can kind of bring in these mitochondria and peroxisomes that are not really functioning anymore. Lysosome, once again, inject some of its digestive enzymes, and we can break those things down so we can reuse the raw materials and not have this stuff clogging up our cells. And lastly, cells have a lifespan. There is a beginning, middle, and end, and towards the end, when things aren't working so well, and that cell needs to go away so we can replace it with a new, better functioning cell, cells have to go through planned death. We call it apoptosis. I cannot pronounce that word very well. Apoptosis. And lysosomes play a big role in kind of that destruction of the old cells. Vacuoles. Vacuoles are basically membrane containers or storage containers. Okay, and they can serve many different functions depending on the cell, as you can see down here in this last one. In plant cells, like we have here, we can see a big plant cell with chloroplast pushed up on the outside. There's the nucleus of the DNA, and a large central vacuole stores water for the plant, but it also helps in maintaining sugar pressure. And we'll talk about this a bit when we talk about cell transport later on in this unit. But if a plant cell, they don't have a skeletal system, right, to keep them upright and rigid. They've got that outer cell wall. Problem is that cell wall is not as strong as you might think. You can think of that outer cell wall like and the vacuole kind of like a bike tire and an inner tube. Okay, the bike tire itself, it's got thick rubber, so it's fairly tough. It can withstand a lot. But if the inner tube inside is not up to pressure, that bike tire is soft and floppy and doesn't do much. The vacuole is like that inner tube. Okay, it needs to have enough water or pressure within it pushing out on the cell wall to keep that plant cell rigid and upright like it needs to be. This is why if you don't water your plants enough, we can't get enough water into the vacuole to maintain that pressure. We'll talk about osmotic pressure later, but we can't get enough water in there, and it's like an underinflated tire, and the cells become floppy and squishy, and your plants wilt. Okay. Animal cells have vacuoles too that store various things. Protists have vacuoles to store stuff, water, food, those kind of things, enzymes, waste, pigments. It all depends on the type of cell and type of organism, what the vacuole is storing, but basically it's storing something. Okay, it's not really doing anything with it, it's just holding it. And it might, excrete some things and push some stuff out of the cell in the case of some protists, but it's basically for storage. All right, and this brings us to how do we put some of these different organelles together? And comes to the point of why we covered the organelles we covered in this unit. There are more organelles. We did not cover all the organelles in cells, but for AP Bio, we want to focus on these major systems and how they work together. So let's take a look at what we call the endomembrane system. The endomembrane system is a system of organelles that function to produce, store, and export biological molecules. Okay, so this is how the cells make what they need to make, store it, and then get it out of the cell. Okay, it's also how we degrade potentially harmful substances. This system includes the nuclear envelope, smooth and rough ER, lysosomes, vacuoles, transport vesicles, Golgi apparatus, and the plasma membrane. We haven't really talked about the nuclear envelope yet, other than things passed from the DNA inside out. And if we're talking about rough ER, we're talking about ribosomes. So we can add ribosomes in there as another organelle. Here we can see kind of the endomembrane system in total. Let's just kind of walk through it. So within the nucleus, we got our DNA. Our DNA will open up, unzip, and you will transcribe an mRNA molecule. The mRNA molecule will then come out and go to one of these ribosomes here on the rough ER. Okay. The ribosomes will then make whatever protein was coded for and materials, and the rough ER will make that. It'll send it in a transport vesicle, either out to do something else within the cell, or more likely send it to the cis face or the near face of the Golgi apparatus, which will then modify and package that, and then send it out the trans face, either to do something elsewhere in the cell like a lysosome, or to send it out of the cell here and get those made materials out of the cell. That is the endomembrane system. That's all of these parts we've talked about working together. It can also make, the endomembrane system will make, vacuoles, which will then store other things, lysosomes, like we talked about before. Smooth ER mainly deals with synthesis of and metabolism of lipids, like steroids, fats, but also detoxification. All right, let's come back now. We talked about energy with mitochondria, right, which takes things like glucose and breaks them down through via cellular respiration to recombine those energy storage molecules into smaller, more usable ones called adenosine triphosphate, or ATP. But where do we get those glucose molecules from in the first place? We get them from chloroplasts via the process of photosynthesis. And like I said before, we've got a unit just on cellular energy, where we'll talk more specifically about cellular respiration and photosynthesis. But let's talk about the organelle real quick here. We need to cover that when we're in this unit. So chloroplasts contain chlorophyll, which makes photosynthesis possible. And it's also a double membrane system like we saw in the mitochondria. Now, within the chloroplast here, we can see we've got the double membrane, and then we've got these little little flat sacs, thylakoid, and we stack them all together, we get something called a granum, okay, or grana. So we got thylakoid, 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 all together now, we got a grana. We can see that here as well, a little closer up. You can kind of think of chloroplast like the solar plant. You can kind of think of the chloroplast as a solar panel of a plant cell. It's going to take the light energy, the radiant energy from light, and through a series of processes, which we'll talk about later, it will produce and store that energy in the chemical bond energy of glucose, okay, which then the plant can do other things with, store it as starch or use it elsewhere. Only plants have chloroplasts, but we reap the benefit of that by eating them, okay, and we'll talk about that when we talk about ecology and how energy moves up the food chain from these producers. So if you have a chloroplast, you're a producer. Let's take a look at some of the structures within the chloroplast a little bit more specifically. The membranes in here, what's important, the membranes contain the chlorophyll pigments and the electron transport proteins that make photosynthesis possible, the full end of it. Now the stroma, this is kind of the liquidy stuff, the space around the thylakoids and granum within the chloroplast. That's got enzymes and maintains a suitable pH for the Kelvin cycle to happen. We've talked about this before, but chemical reactions are very picky, right? You need to have the right temperature range for them to happen. If we get too hot or too cold, it doesn't happen. If we don't maintain that kind of homeostasis, and various different chemical reactions within the cells need different pHs. So in the lysosome, for instance, I didn't mention this before, but within the lysosome, in order for those hydrolysis reactions to occur, they occur best at a slightly more acidic environment than the rest of the cell. And the same thing here, we need the right pH for the Kelvin cycle to happen within the stroma. So that's one of the compartmentalization parts of chloroplast, is maintaining that right pH within them, okay, for the Kelvin cycle. And then we have the thylakoids here, okay, have all the right kind of enzymes, synthase, ETC and ATP synthase, that's an enzyme, for photophosphorylation, which will happen in them. We'll talk about them later. And the granum, these flattened sacs, that increases the surface area to volume ratio, okay, so that we can um, That's what I'm looking for. So we can concentrate these reactions, and this is where light dependent reactions occur. There are both things we call light dependent and non-light dependent or dark reactions in photosynthesis, which we'll talk about later. But the light dependent ones happen in these granum and the thylakoids here. Okay. And that is it for talking about 2.0, the cell theory, and 2.1 and 2.2, the structures and their functions within the cells.